Hello students, the experiment that we are going to study about today is about micropropagation. The aim of today's experiment is to study about micropropagation and hardening or acclimatization of regenerated plants. The equipments required for today's experimentation are autoclave, pH meter, magnetic stirrers, magnetic beads, weighing balance, laminar airflow, etc. The materials required for the experimentation include reagent glass bottles, spatula, tissue rolls, distilled water, cotton plugs, aluminum foils, scalpel handles, sterile blades, forceps, borosel glass test tubes of the size 115 to 25 mm and preferably without rim, containing Murasige and Skook's 1962 medium, marker pens and fresh shoot tips of the plant Strobilanthes cusia. Let us now see the theory of micropropagation. Micropropagation or in vitro clonal propagation is a rapid method for vegetative or asexual multiplication of genetically identical copies of a cultivar using tissue culture technique. Using this technique, in vitro clonal propagation can be achieved in a short time in a small space and large numbers of plants can be produced starting from a single individual. Micropropagation can also be used for production of virus-free plants by culturing the apical meristem of the plant. The process of micropropagation can be divided into the following stages. The first is the stage 0. This is the initial step of micropropagation in which stock plants used for culture initiation are preconditioned before culturing in vitro. It is usually done by growing the stock plants under carefully monitored conditions as at a relatively low humidity for at least 3 months and also include measures adopted for reduction of surface and systemic microbial contaminants of the explants. The next is the stage 1 or the explant culture initiation. Murasige defined this stage as the first in vitro stage for micropropagation, and the main steps involved in this stage are preparation of the explants including surface sterilization and initiation of culture. Surface sterilization of the explants includes procedures to reduce surface microbial contaminants and also to control microbial contamination within explant tissues which could not be eliminated at stage 0. For micropropagation, explants with preformed vegetative buds such as shoot tips and axillary buds which are suitable for enhanced auxiliary branching are usually selected for culture initiation. Initiation of cultures involves culturing the explants on suitable culture media followed by subculturing a few times to see whether the microbial contaminants are fully removed or not. The next stage is the stage 2, which is the multiplication of microshoots. This stage is the major stage of micropropagation process and involves the multiplication of shoot buds using defined culture medium that stimulates maximum proliferation of microshoots. A growing explant can be induced to produce vegetative shoots by including a cytokinin in the medium. A cytokinin is a plant growth regulator that promotes shoot formation from growing plant cells. The next stage is stage 3 or the rooting of microshoots. The in vitro regenerated and multiplied shoots produced in stage 2 are transferred to a rooting medium in this step to obtain complete plantlets. The media used for rooting normally contains less salt concentration and includes growth regulators like auxins, which promotes rhizogenesis in the microshoots. Sometimes shoots of certain species may be directly established in the soil and root them in vivo or ex vitro like microcuttings. In such cases, stage 3 is usually skipped. The last stage in micropropagation is stage 4, which is the acclimatization or hardening of. It is the last step of micropropagation and comprises steps taken to ensure successful transfer of the plantlets of stage 3 from the aseptic culture environment of the laboratory to the environment of greenhouse, and the process is known as acclimatization. Since the plantlets growing in the culture vessels were growing under 100% humidity inside the vessels, and the regenerated plantlets usually have no wax and poor stomatal response, they need to be acclimatized to ensure successful survival outside the vessels. Therefore, acclimatization is carried out by supplying bottom head aids to pots with plantlets or cuttings and maintenance of a dense fine particle fog system within the greenhouse to enhance the rooting process. The plantlets can also be established in the artificial growing media such as soil-less mixes, rockwood plugs, or even sponges. A growing rooted shoot can be removed from tissue culture and placed in soil. When this is done, the humidity must be gradually reduced over time because tissue cultured plants are extremely susceptible to wilting. The general process of micropropagation involves the following steps. The first step is the selection of healthy plant material explants like the shoot apical bud from a healthy mother plant. The next step is surface sterilization or washing of the plant material with detergents, sterilants, and antibiotics to remove microbial contaminants. The third step is media preparation and sterilization. The medium can be solid or liquid, and a particular medium is selected for a plant after trial and error since the requirements of each plant species or sometimes variety within a species vary depending on the genotype. The fourth step is inoculation or transfer of explants to the medium. The cultures are then incubated in culture rooms for shoot multiplication and root induction. The fifth step is the hardening or acclimatization. It involves the transfer of a growing rooted shoot or plantlet from test tube or flask to pot containing soil and maintained at high humidity, and humidity is gradually reduced during this process since the tissue cultured plantlets have no waxy cuticle to prevent water loss. The final step is the transfer of hardened plantlets to field or natural environment. Let us now see the procedures for micropropagation using shoot tip explants of Strobilanthes coccia. The first step in the process is to collect healthy shoot tip explants of Strobilanthes coccia. Next, we should rinse the shoot tips under running tap water and then put the plant material in a jar with screw cap lid containing water and wash with 0.1% detergent for 3 to 4 minutes. Now gently agitate the content of the jar every 20 to 30 seconds during the washing step. Next, pour off the detergent and rinse the plant material with tap water and cap the container again. Now spray the container with 70% ethanol and bring it to the sterile workbench of the laminar flow. Spray the sterile workbench of the laminar flow with 70% ethanol and also spray vessels and all tools packets containing petri dishes, forceps, scalpel handle, razor, blades, etc. with 70% ethanol before placing them on the work surface. Now remove the cap of the container containing the plant materials and pour 0.1% mercury chloride solution and replace the cap. Wash the plant materials by intermittent sacking for 6 minutes. Next, pour off the mercury chloride solution and pour sterile water over the plant material and rinse the plant materials for 2 to 3 minutes by gently agitating. Repeat the rinse with sterile water for 3 to 4 times. Next, using the sterile forceps, transfer the shoot tips in a sterile petri dish and cut off the damaged portions of the bases using a sterile blade. Now open the cotton plug of the culture tube containing MS vessel medium and flame the mouth of the tube. Next, flame the forceps and cool them by touching the medium in the culture tube. Now use the forceps to hold a shoot tip explant and place it on the medium by gently pressing so that the explants make contact with the medium. Now replace the cotton plug of the culture tube and mark the tube with date and details of the experiment. Incubate the culture tubes in a growth chamber provided with 16 hours light and temperature maintained at 25 plus minus 1 degree Celsius for 3 to 4 days. After 3 to 4 days, look for signs of contamination during this period. If contamination appears, fresh cultures can be inoculated by following the above same procedures. Next, if no contamination appears, then transfer the explants on medium containing higher cytokinins to oxygen ratio for shoot multiplication. Since naphthalene acetic acid or NAA and kinetin were found to be suitable for shoot multiplication in this plant species, let us culture the explants on MS medium supplemented with 1 mg per liter NAA in combination with 1 mg per liter kinetin. After inoculation, incubate the culture tubes in a growth chamber provided with 16-hour light and temperature at 25 plus minus 1 degree Celsius for 5 to 6 weeks. Observe the cultures every week. After about 2 weeks, the shoot tips may start proliferating shoot buds and multiple shoot buds will be visible within 5 to 6 weeks. The shoot buds may be separated, trimmed and subcultured on fresh culture tubes containing shoot multiplication medium and cultured again for generating sufficient number of shoots. Once sufficient number of shoot buds has been generated, the shoot buds are separated, trimmed and transferred to flask containing MS medium supplemented with higher oxygen to cytokinin ratio for root induction. The flasks containing the cultures are kept on racks inside a growth chamber provided with 16 hours light and temperature maintained at 25 plus minus 1 degree Celsius for 5 to 6 weeks. After about 2 weeks, the roots may start proliferating from the bases of the shoot buds and efficient rooting will be visible after 5 to 6 weeks. The 6-week-old rooted plantlets are ready for hardening and transplantation. The rooted plantlets are gently taken out from the flask and their roots are washed off with tap water to remove any trace of agar sticking on them. The plantlets are then transplanted in perforated paper cups containing 1:1 ratio of sand and soil and kept covered with clear polythene bags having a few holes on them for the initial 10 days for hardening. The plantlets are maintained in a 50% shaded net house under high humidity for 10 days and the polythene bags were finally removed. 4-week-old hardened plants were then transplanted to bigger earthen pots or to the field. Let us now see the results of the experiment. The shoot tip explants proliferated multiple shoot buds on bud induction medium after 4 weeks of culture. About 11 shoot buds were obtained from a shoot tip explant cultured on medium containing 1 mg per liter NaA in combination with 1 mg per liter kinetin. The shoot buds cultured on this media also sought the induction of rooting and further multiplication was achieved by subculturing the buds on this media after every 4 weeks. The in vitro regenerated plantlets showed 70 to 80% survival during transplantation. Thank you.